Variation on survival and immature development of Ceratitis capitata population rather than two key overwintering hosts. Well, Nikos, please.
So, good morning, everybody. So, I was actually about to quit my talk. I said first that I feel like an invasive talk or an invasive species in this session with all this taxonomy, and then after this great performance of Luke, I said, what can I say to bring your attention to my talk? But anyway, so my talk is completely different of what you have heard. It's not about many species, about discoveries. It's about a species that we know it very well. It's medfly, and it's about the invasion process of medfly. We all know that this is a really important species that this is not working. Okay. So, and because of all these human activities, like the commercial trading and also the people movement, medfly has dispersed from the ancestral habitats of sub-Saharan Africa to almost all over the globe. First in the Mediterranean region, then from the Mediterranean to the New World, to Australia, to several other places. But what we have seen recently in Europe, there is a gradual dispersion and expansion of the geographic distribution of medfly to the north. So in Europe we don't face problems only with medfly, but we have also some additional invasive species. We have the Bacteroceratidae dorsalis, the oriental fruit flies, and all the oriental fruit fly, and also the peach fruit fly that are more frequently, let's say, they are detected in European countries nowadays. And last year there was a major outbreak of Bacteroceratidae dorsalis in Italy, in the area of Napoli, which seems to be quite serious. But my talk focuses on medfly and its dispersion from the Mediterranean coasts to the northern, to the central European countries. So it seems this is gradual, it's happening, and it seems it's also quite serious, and it attracts the attention of several recent studies. Actually, as you can see, there are studies that they examine the detection of medfly in countries like Austria, Germany, northern Italy, and in many places in France. And there is some modeling that predicts that some of the habitats in northern coasts of the Mediterranean, but also in Central Europe, they are becoming suitable for the establishment of medfly. In fact, we have a recent study that it's by Sinisevska and colleagues that working with the climates modeling predicts that, not predicts, actually we are proving that some of these habitats here in yellow color, from unsuitable, they become suitable from the 70s to the last decade. So it's after all this thorough climatic analysis, it seems that there are these changes, and we have clusters of this yellow color as you see in Central Europe, that it's kind of suitable for the development of medfly. And if we look at this graph here, this, you see all these are detections in France and in northern Italy, in Austria, in Ukraine, here in the Balkan Peninsula. There are many detections, and there is a recent study with several detection and analysis from Germany. So that the conclusion is that the fly cannot survive there, but there is some structure in the population that may show some, breeding and some existence in the area. So, but for medfly to survive in these cooler environments, it needs to overwinter. This is the most important thing. It needs to go through a long period of harsh conditions and also a long period of absence of host fruits. Consider that in these countries in Central Europe, you don't have citrus and you don't have fruits from the end of the season, from October, let's say, until next June, like next May in the best conditions. So Our question was for several years how mayfly can survive in these harsh conditions and how can overwinter. And actually, this was part of my PhD thesis to see and to study the overwintering of mayfly in northern Greece. And northern Greece is quite cool, the area of Thessaloniki, because it receives all the, let's say, cool air from the Central Europe. So what we have shown in these studies is that mayfly overwinters inside the infested fruits that are the last to be infested in autumn. It's apples in this area, and the larvae can continue developing in apples until April, from November, October, November, all the way to April. So, and that is the overwintering stage in this study. In other studies in southern parts of Europe, it's larvae within bitter oranges, and apparently as you go further south, you have all the stages that can survive during the winter. So, our question here, our aim was to explore the overwintering capacity of different mayfly populations into overwintering hosts. So we considered both, we considered different mayfly populations. We have 6 populations from the southernmost point of Europe all the way to Austria. In Austria there is a breeding population for the last 10-12 years, and the guys, they collect infested fruits every year and they keep a colony. So we managed to bring these flies to my laboratory. Also we got flies from Croatia and 4 Greek populations. And what we did was to run a common garden experiment, having these 2 overwintering hosts, the apples and the bitter oranges, and then look at the development at 3 temperatures, at 15, 20, and 25. So it sounds like a simple, straightforward experiment, but it's really laborious, considering the different populations and also working in lower temperatures that you know, the fruits do not respond very well. So what we did is to inject eggs, to place eggs in the flesh of the fruit, and then to put these fruits in some plastic trays over dry sand and to take them to the incubators, to the different temperatures. And we were recording the incubation every day until the end of the last individual that managed to incubate. So, first we try to analyze the climatic indices. We used 90 climatic indices, and we followed this PCA analysis to group the different areas. So we have some nice grouping here in this graph, as you can see. It's first we have the 2 islands. These are the 2 islands, the Chios and Chania. This is Crete. Then we have Vienna and Zadon, the northern populations. And here is more continental Greek populations from Volos and Thessaloniki. And the PCA, the higher the PCA1, the warmer the temperatures and hotter and drier the summers. In PCA2, the higher the PCA2, the higher the temperature variation, the fluctuation of temperature, and the higher the winter precipitation. So that these 2 principal components explain more than 80% of the variance. And if we look at the results of these studies, we have here the survival rates of the different populations from the northern to the southern population. And you see, we can not really make a lot of statements out of this graph. But as a general conclusion is that all populations do not perform very well at low temperatures. And these are for various reasons. And then you can see differences that are related to the host, and then there are population differences as well in survival rates. So, but we looked also at the developmental duration. In developmental duration, consider that this is really important for the overwintering, because if it's to overwinter within the fruit, it needs to extend the larval stage to several months, to many months actually. So, and as you can see in this graph, we have extension of the immature development of well over 100 days, and in some of the populations we have some high variability, some plastic adjustment in the lower temperatures, and in all temperatures we have some variability, but this variability you can see it in these Greek populations mainly, but not in the two northern populations. And what else is important is if we look at the extremes here, we have some larvae that at 15 Celsius in the laboratory, they left the fruit to pupate after 4 months. So this hue of the distribution is really important for the survival and the overwintering of medfly in these northern cooler countries that the winter is much longer apparently. So also we ran a model and we included the PCA as one of the predictors of the larval development in this model, the PCA1 and the PCA2. Both of them, they were significant predictors of the larval development, and this is the density plots. What is interesting in this graph is that we mark the different populations according to the PCA. This is the treatment, the temperature that the fruits were exposed. You cannot make much out of this. You need the details. And here is all the populations from the southern one to the northern population that we have considered in this study. So you can see the differences in the developmental duration. But what is really interesting in this study is to see that these three populations, they have a much broader plasticity in the developmental duration, which is missing from the northern populations. And this is really an important finding of this study. Also, if we look at the PCA2, again, it's the same data, but we have a different color because of the PCA2. Again, we see that these central, let's say, continental Greek populations mainly, and the Chios Island, they manage to extend and to plastically respond to the lower temperature and to the different fluids. So the overwintering capacity differs among the different populations. So, and to conclude the results of this study is that first we've seen that survival and development at both overwintering hosts and lower temperatures vary a lot among the populations. And it seems that older invaders express higher levels of developmental plasticity than the northern populations. Consider that medfly is established in the Mediterranean area for more than 100 years, and some of these northern populations, they are for a few decades, and in the Austrian population, it's not sure whether it's permanently established or not. So, and overall, this is the microclimate of the region of origin, and the invasion history seems to determine the invasion success of medfly in the northern European countries. So, and this work is supported by two projects. The first is the FFIBM project. It's a Horizon European project that is in its last year. It involves 21 participants from different countries. And also the project, the medfly project, that is funded by the Hellenic Foundation for Research and Innovation. And we are collaborating in this project with the group of Daniel Hahn from the University of Florida, Gainesville, and also from the group, with the group of John Terblans from the Stellenbosch University. So that's it, thank you very much. And if I have just a small minute, just to drag your attention to another project that we run in Europe. It's called REACT. It started last year, and the aim of this project is to find solutions for the invasive species, again targeting the two Bactrocera species, the Bactrocera dorsalis and Bactrocera zonata. So thank you very much.
Do we have Al, please? Yeah. We have time, yeah. Let's talk a little bit short.
That's very interesting. I was wondering, is there much export of fruit out of northern and central Europe, like happening with quarantine?
Yeah, but yes, indeed, there is a lot of fruit export, considering pome fruits mainly, apples, and probably some prunus. Yeah, there is this activity, and in fact, the range expansion of medfly to these countries, it's actually poses a huge risk to all this fruit production industry. For example, in northern Italy, they are producing apples, mainly apples in the area of Trentino, and they had established a very nice IPM project against the codling moth. But now with medfly, even this IPM project is under a threat because they cannot execute it. They need to spray close to harvest. And I suppose there will be some issues with exporting fruits from these countries if, medfly permanently established there.
I wonder, maybe I missed it, is the kind of distribution of environmental, temperatures that each of those populations experiences, does that match what you see in terms of the development time?
Yeah.
Do the central Greek populations, do they have a wider distribution of environmental temperature extremes compared to Crete or in the north?
Yeah, it seems that let's say the continental, the central Greek populations, the microclimatic conditions indicate that there is a greater fluctuation of the temperatures. And this is a seasonal fluctuation, but it's also diurnal fluctuation. And this might be one of the reasons that they have adapted to extend more this larval stage. So it seems that there is some adaptation. We are still working on the genetics of the different populations.
You had a question, right?
Just curious to know if different haplotypes and
Yes, Miguel, indeed, we are actually analyzing the data. I have not yet the data, and we have included not only these populations, but some populations from Israel as well. We have a really good transect of the latitudinal transect of the populations that we analyze. And in collaboration with the Stellenbosch University, we are working also in the southern hemisphere, from Kenya to the Cape Town, let's say, and we are analyzing this data as well. So at the end, I suppose having all these latitudinal populations analyzed in the genetics and bringing this biological data, we might have a better understanding of the traits that make medfly such a successful invasive species.
And these acute hot temperatures, it's going to be 45 in Spain this weekend. I guess you will have to start studying the acute effect of these short hot spells.
Actually, we have studied these effects. We have studied the critical thermal minima and critical thermal maxima for the different populations, but we can report it in another meeting, I suppose. Okay. Thank you very much.
Thank you, Nikos.